Did you know that 90% of oxygen you breathe is used in one tiny process happening in your cell? That process is the electron transport chain. In this presentation, we'll explore how the ETC works, its key players, how it generates energy, and why it's vital for life. The ETC is a series of proteins in the inner mitochondrial membrane, and it consists of specific mitochondrial proteins and enzymes. Some examples here are NADH reductase and cytochrome reductase. The process transports electrons down the ETC to pump hydrogen ions into the inner membrane space, creating an ion gradient. The process is split into two parts. After coupled redox reactions, ADP is phosphorylated into ATP through a process called chemiosmosis. A total of 4 net ATP is produced so far through glycolysis in the Krebs cycle. This amount is not enough for life to function, so the ETC is used to pick up some of the slack. Molecules of NADH and FADH2 account for the energy taken from every glucose molecule. NAD+ and FAD+ are reduced to form NADH and FADH2. These molecules are electron carriers, which takes the electrons down from the Krebs cycle to the area of oxidative phosphorylation. The end goal of the ETC is to generate an electrochemical gradient to produce ATP. It's also to use NADH and FADH2 to produce ATP and form water as a byproduct. Listed below is the entire respiration equation. These first 4 protein complexes are involved in the oxidative part of oxidative phosphorylation, and ATP synthase is involved in the phosphorylation part, where ATP is being phosphorylated. Keep in mind that the names for all these protein complexes don't need to be memorized for the AP exam. In eukaryotes, the ETC is embedded in the inner membrane of the mitochondria. In prokaryotes, these molecules are located in the plasma membrane. The folding of the inner membrane to form cristae increases the surface area. This beautiful structure provides many copies of all the proteins and enzymes to increase their function. This example is directly how structure resembles function. An example of these proteins and enzymes that could be used are prosthetic groups. These are attached to the mitochondrial proteins essential for catalytic functions. This structure is well suited for the series of redox reactions that take place between certain protein complexes.
When does ETC take place? Oxidative phosphorylation is the last step of cellular respiration. At the end of the first 3 processes in cellular respiration, which are glycolysis, pyruvate oxidation, and the Krebs cycle, FAD+ and NAD+ are reduced into NADH and FADH. Oxidative phosphorylation is the cellular respiration's last stage or step. It's what produces the main products. The first 3 steps help create the molecules needed for phosphorylation phase. During the phosphorylation stage, redox reactions occur due to the electron transport chain, and ATP is synthesized by chemiosmosis. The electron transport chain is a group of protein complexes that receive FADH and NADH and oxidizes them. As the protein complexes in ETC are reduced, energy is released. These electron carriers are the major energy-containing carriers, as they contain not only electrons needed for the reaction, but also the energy used to make a proton gradient, which will be mentioned later.
In the first complex, NADH will initiate the electron transport chain through transferring electrons into flavin mononucleotide, or FMN. Afterwards, electrons are then transferred towards iron-sulfur, or FeS clusters. The final electron acceptor of complex 1 is called ubiquinone, or also called Q. As this process occurs, energy is released via the translocation or movement of hydrogen ions from the mitochondrial matrix to the intermembrane space across the inner mitochondrial membrane, which contributes to the formation of a proton gradient. NADH is considered to be more effective than FADH due to the fact that NADH allows for an increased amount of ATP energy production, as NADH enters the electron transport chain at an earlier stage, which allows for more protons to be pumped across the membrane. Now, moving on to the second complex, FADH will initiate the electron transport chain by transferring electrons into another iron-sulfur cluster. The electrons will then be transferred into the next acceptor. An important thing to note is that the second complex will not pump protons across the mitochondrial membrane. Keep in mind that this ties back to the fact that NADH is going to be more effective in regards to ATP production. To summarize, electrons are being transferred across the electron transport chain, as it pumps protons across the membrane in which it creates a proton gradient. Now, what is our outcome? Both the NADH and FADH2 are transferred between complexes 1, 2, 3, 4, and 5, in which the fifth complex is also known as ATP synthase. Oxygen is the final electron acceptor of these protons and will create a byproduct of water. The proton gradient will also allow for the ATP synthase membrane to produce ATP energy.
Does it end here? Oxidative phosphorylation doesn't just end after the ETC. However, the results of the ETC help set up for the next process, chemiosmosis. While redox reactions take place in the protein complexes, energy is released. This energy allows for hydrogen ions to be pumped out into the intermembrane space through active transport, creating a proton gradient. There would be a higher concentration of hydrogen ions in the intermembrane space rather than the matrix. These hydrogen ions are then transported into the matrix through ATP synthase, allowing for the creation of ATP.
Something that might be surprising about the electron transport chain is that it doesn't directly make ATP. So, how does it ease the transport of electrons and release said energy into smaller and more manageable amounts? Well, we can think of a process called chemiosmosis for ATP synthesis. This process couples the electron transport and energy release produced by the ETC by actively transporting H+ ions across the inner membrane of the mitochondria. Energy is stored in this pre-existing hydrogen ion gradient, with a higher concentration of H+ ions in the intermembrane space than in the mitochondrial matrix. This difference in H+ ions also results in a difference of pH across the membranes. Overall, there are 4 main steps when it comes to the production of ATP during chemiosmosis. Firstly, the hydrogen ions move down the gradient one at a time and enter a channel in the stator in the mitochondrial membrane. From there, the ions bind with binding sites in a rotor, spinning in the membrane. This rotor turns one full rotation before expelling the hydrogen ions into a second channel in the stator and into the mitochondrial matrix. Finally, the rotor spins, causing its internal rod to also spin and activating catalytic sites in the knob below it. This causes the binding of ADP to phosphate, ultimately creating ATP energy. Now, after this whole process, aerobic respiration is complete. If there's any leftover oxygen that is produced, cellular respiration takes place and transfers energy from one molecule of glucose into 33 ATP molecules, which is also stored chemical energy. During this, CO2 and H2O are released as waste. Delving deeper, where does the wastewater come from? Well, the ETC issues cytochromes, which are redox-active proteins that contain heme and assist in electron transport. One of the cytochromes, side A, passes its 2 electrons to oxygen, which we know to be extremely electronegative. Each oxygen atom attracts a pair of positive hydrogen ions, neutralizing the negative charge of the 2 electrons and binding the H2O, also known as water. Now for a quick concept check on chemiosmosis. How does the electron transport chain create a hydrogen ion gradient during chemiosmosis? Pause the video.
Hey guys, so now we're going over specifics of the electron transport chain involving uncoupling agents. So during normal energy production, our energy organelles will convert chemical energy or solar energy into ATP. However, with the introduction of an uncoupling protein, the transport of protons across the inner mitochondrial membrane is stimulated. This process decouples the electron transport chain and ATP synthesis, weakening or dissipating the electrochemical gradient and the proton gradient. If we take a look at the image on the right, we can see that the flow of electrons through each complex creates a proton gradient across the inner mitochondrial membrane. However, with this uncoupling protein, or UCP1, protons flow back into the mitochondrial matrix and collapse the proton gradient. This specific process is called thermogenesis, where heat is generated instead of producing ATP, And for the most part, this is considered a trade-off because while our body temperature is being maintained, it's at the expense of ATP production. So as a result of uncoupling agents disrupting ATP synthase, we get what is called mitochondrial uncoupling. So this process defines the loss of the normal energy production in the mitochondria as ATP production is bypassed because the potential energy is released as heat. The image on the left defines the whole process, which we can split up into three components. Our first part involves oxidative phosphorylation, which was already mentioned some slides back, but just for a brief review, electrons from NADH and FADH2 move along the electron transport chain, and the products of this process will be ATP and H2O. Then our second step is called reactive oxygen species generation, and this is when electrons leak from the electron transport chain and form molecules like superoxide and hydrogen peroxide. At low levels, this process plays a part in cell signaling, but at higher levels, it can cause oxidative stress, which is an imbalance between the production of damaging ROS molecules and the ability of a biological system to detoxify these products. As a result, mutations in our DNA and misfolding of proteins lead to neurodegenerative disease and heart disease. Our last and final step is the mitochondrial uncoupling. And basically, the uncoupling proteins decrease ATP levels in the mitochondria because these agents allow protons to completely bypass ATP synthase. So this process is very detailed and has a lot of information, but I like to think of it as a car being turned on in the winter. Essentially, the car engine is a mitochondria and it burns fuel efficiently, but the energy is dissipated as heat instead of being converting into mechanical energy or ATP. As a result, the car gets warm and protects you from the freezing temperatures outside the car. So before I move on to the next topic, we'll do a quick multiple choice question to check our understanding of this topic. So you can read the context if you want, but the question asks, which of the following statements provides reasoning that supports a claim that brown fatty tissue keeps an animal warm? So you can pause the video if you want to read the answer choices before I go over the answer by 2 seconds. So the correct answer choice happens to be option B, and this choice is correct because the uncoupling proteins cause a reduction in the proton gradient across the inner mitochondrial membrane as protons constantly leak into the matrix. Heat energy is released as the protons move, but less ATP is made because of the decreased concentration of the hydrogen ions available to move across ATP synthase. Now, our last two slides are going over weight loss, starting with our two weight loss hormones, leptin and ghrelin. So leptin is a hormone produced by the adipose tissue or your body fat, and it sends a signal to the hypothalamus, which is a part of the brain that controls autonomic functions. High levels of leptin decrease appetite and increase energy expenditure, while low levels do the opposite. On the other hand, we have ghrelin, which is the exact opposite of leptin. So ghrelin is secreted by our stomachs, and when we fast, it undergoes a similar process of signaling the hypothalamus, but at high levels, it instead increases appetite and stores energy for future use. Despite the differences, leptin and ghrelin influence energy balance and metabolism, indirectly affecting mitochondrial uncoupling by modulating appetite, fat storage, and energy expenditure. Now, this last slide is going back to our uncoupling agents, and studies from the past 20 years have been going over different solutions to weight loss. One particular solution that was considered were artificial uncoupling agents, but they quickly turned out to show fatal side effects when used. Some examples of these synthetic uncouplers included DNP and FCCP, which both act as protonophores or proton translocators that can disrupt ATP synthesis. And that's it. These are citations.